Okay, good afternoon, everybody. Welcome to this session on, I've forgotten the title of the session, the promise of nucleic acid-based therapies in CF, prospects and progress. I'm delighted to be working with my co-chair here, Dr. Deepika Polanini from St. Louis, who you'll all know. We've got four great speakers, well, three of myself, this afternoon. We're going to get straight down to the talks. The four talks, we've got Professor Batsheva Karem, who's going to be talking about antisense oligos. And in many ways, this is the one which is closest to the clinic. Jane Davis then from London will be talking about progress with lentiviruses. I'll be talking about gene editing, and Deepika will finish off this session on real-world challenges and perspectives. Each speaker will be talking for about 20-22 minutes, and then we'll have a few minutes for questions. If you could come up to the mics for this, because the session is being recorded, and we want to capture the questions as well for the people who are listening online. We've also got an iPad here. We're very high-tech. We may even get some questions from people from far, far away. Okay, I'll not carry on anymore. If I can operate the system with help from Bill over in the corner there, and introduce the first speaker, Batsheva Karem. Thank you.
Hi, good afternoon, and thank you for coming. It's a competition. There are many other presentations in parallel. So thank you, the organizers, for inviting me to present our progress on antisense oligonucleotide therapies in CF. And here are my disclosures. I am the Chief Scientific Officer and the scientific founder of SpliceSense. So antisense, they are the focus of the presentation. They are single-stranded RNA-like short sequences, about 20 nucleotides, that can bind and modulate RNA splicing. And they are specific to the target mutation region and thus potentially avoid off-target effects. They are chemically modified for stability and increased cell uptake, and they are clinically relevant. As of today, there are 12 clinically approved ASO on the market for other diseases, including genetic diseases. So here is the pipeline of SpliceSense and the various projects that we work on. The first one that I will focus on today is targeting a mutation called 3849, which I will explain in a moment. Another project is targeting the nonsense mutation W1252X. And there are two additional projects aimed to target mucin proteins generated by MUC5AC and MUC5B, and they are relevant for pulmonary diseases, including CF. So the 3849 mutation, the clinical features are interesting. The sweat chloride, and the data are from CFTR2, updated this month. So the sweat chloride of patients carrying the mutation is lower, 65, compared to the entire CF population. The patients are older, and their pancreatic status is different from the entire CF population. Most of the patients are pancreatic sufficient. However, their lung disease is comparable in severity to that of all CF patients. We can see here that the Pseudomonas infection rate is similar between 3849 patients and others. And FEV1, the lung disease in patients carrying the mutation, is variable, similar to the variability we see in the entire CF population. More recently, Meir Meizahav, Eitan Kerem, and others analyzed data from the ECFS patient registry and found, as can be seen here, that people with CF carrying the 3849 and F508del here in blue have comparable FEV1 rate of decline to that of homozygote in black, homozygote F508del. So patients suffer patients with this mutation suffer from a severe disease. The 3849 is a splicing mutation, and I would like very briefly to review splicing. So as probably you know, the vast majority of the human gene comprised of coding sequences, the exons, separated by non-coding sequences, the introns, and splicing is the process in which introns are removed from the pre-messenger RNA such that the exons can accurately be joined together. Splicing is mediated by many proteins, over 150 of them, and they are mediated also by sequences. I will mention the donor sequence, GU, and the acceptor, AG, and the pyrimidine, etc. The 3849 mutation is a C to T change in intron 22. And the C to T change generates a donor splice site, a cryptic one. And this is activating the acceptor site, cryptic acceptor site, 84 nucleotide upstream of the mutation. And this results in the recognition of these 84 base pairs as an exon. So this cryptic exon is spliced into the transcript. And unfortunately, in frame, these sequences carry a stop codon. So due to this, the transcript that are including this cryptic exon are degraded by the nonsense-mediated messenger RNA decay surveillance mechanism, and as a result, no protein is produced. However, in many patients, some normal splicing also occurs despite the mutation. So many years ago, on a small number of patients, we found a very strong correlation between the level of correctly spliced transcript and disease severity measured by FEV1. I would like to note that the current CFTR modulators are approved for patients carrying the mutation, and the drugs can only affect the correctly spliced transcript, so which are variable among patients. So our idea was, based on the data that I showed, that modulation of splicing, increasing the correct splicing, may affect disease severity. And the approach in order to increase correct splicing, we are using antisense oligonucleotides. So the rationale is that the ASO will bind to the cryptic exon and will mask it so it will not be recognized as an exon, and as a result, there will be no aberrant splicing, but only or mostly normal splicing. And I would like to emphasize that normal splicing will generate a normal protein, which should be functional as the wild type, since the cryptic exon is not there. So in SpliceSense, we developed an algorithm to design antisense oligonucleotide. The algorithm enables us to identify splicing motif within the target sequence, to design efficient and specific binding ASOs, and also the algorithm takes into account safety and immunogenicity. So for this project, we designed the ASOs that we see here, covering the exon in black and the flanking sequences. And the first level was to analyze whether indeed treatment in cells with ASOs that we designed can change the splicing pattern, can increase correct splicing. So the first analysis was done in FRET cells, flipped in with the CFTR cDNA carrying either the wild type sequence. So you can see here a gel of RT-PCR. All the products are correctly spliced as expected. And we can see here that control, that by by expressing the 3849 allele in these cells, all the RNA is aberrantly spliced, like in the very, very severe patients. And I'm showing here the result, the effect of the antisense on the splicing pattern for several antisense oligonucleotide. All of them, in this example, show very high level of correct splicing, starting from only aberrant to high level of correct splicing. So the next question was, can these transcripts be translated to normal, full-length, and hopefully functional CFTR proteins? And here are results on Western blot. Here are the FRET cells. We can see that treating the cells with control ASO, there is no CFTR protein. And following treatment with several of the ASOs, we can see here mature CFTR proteins, BNC. We see it in another system as well. So our ASOs form full-length mature CFTR proteins, which shows at this moment this ASO, SPL84-23, that I will call from now SPL84 for drug development. The next step was analyzing the effect of this SPL84 on the CFTR function. And this time it was done in primary respiratory epithelial cells derived from patients carrying the 3849 mutation. Here is an example. The experiment were done in collaboration with Isabelle Sermet. And we can see here a trace in Using chamber. So if the cells were H&Es, human nasal epithelial cells, derived from a patient homozygote for the mutation. So treating the cells with a control irrelevant ASO, there is no function. Following the treatment, we can see the activation and the inhibition with a specific inhibitor, a very nice trace. We extracted RNA from the filters that were analyzed in the Using chamber. And we can see here that with control ASO, the baseline is the level of correct splicing is very, very low, which explains no function. However, following the treatment with SPL84, the majority of the RNA is correctly spliced, so almost complete correction of the splicing defect. And here is a summary of repeated experiment, both in human bronchial epithelial cells and human nasal epithelial cells, treating the cells with SPL84. So this is the homozygote cells that I've shown an example. So these are repeated, an average of repeated experiment. And we can see that the CFTR function reaches 90% of wild type. Here are the results in HBEs from a patient heterozygote for the 3849 mutation. And we can see that in this heterozygote, we are trying to correct one allele, and the level of wild type activity is close to 50, around 45% of wild type. And last is the results of in H&Es derived from 5 different patients, all of them carrying one 3849 allele, and the results are the same as in HBEs, over 40% wild type activity. So to summarize this part, SPL84, which is targeting the splicing mutation, corrects the splicing defect, generates full-length normal CFTR proteins, and rescues the CFTR function, leading to almost wild-type activity. Splicins end products are inhaled drugs. So this enables direct non-invasive delivery, even of high doses of drugs, with minimal systemic absorption. And this is very important for antisense oligonucleotides, since systemic exposure of them might be toxic. Patients might develop thrombocytopenia, etc. And another point that I want to emphasize that the drug is given to patients once a week, so potentially reducing patients' treatment burden. All these results and more that I don't have time to share today led us to initiate clinical trials. So we already successfully completed phase 1, which was a single ascending dose in healthy volunteers done in Jerusalem, in Israel. And the results showed that there are no significant adverse event or significant related adverse event. Thus, this is a promising safety profile. And systemic exposure with SPL84 was low, as we predicted and hoped, and was dose dependent. This led us to initiate phase 2, which is a placebo-controlled, multiple ascending dose trial in subject with the 3849 mutation, of course. And the primary objective of this clinical trial, phase 2, was to first evaluate the safety and tolerability of multiple ascending doses of SPL84. And the secondary objective was to assess preliminary efficacy of FEV1 of multiple ascending doses of SPL84. So earlier this month, Splicens announced positive results from our ongoing phase 2 study. These are interim results with our lead ASO, SPL84, for patients carrying the 3849 mutation. And the key highlights of these preliminary interim results are that there is a strong safety profile with no safety signal identified as of to date, and there is meaningful efficacy. The lung function improvement observed in 70% of the patient was with a mean absolute FEV1 increase of 10% relative to placebo. And if we compare the increase within each patient to baseline, the increase is at least 5% compared to baseline. So as far as we are aware of, this is the first evidence of potential clinical benefit of antisense oligonucleotide therapy in a pulmonary disease. And this is encouraging us to bring into the clinic other programs that we have, including the W1252X mutation in CF. I would like to end my talk by raising a question and show some results. And the question is, would a combined treatment of SPL84 and CFTR modulators may lead to additional benefit to heterozygote patients carrying the 3849 mutation and F508del or other mutations that are responsive to the current available modulators? Can the treatment be beneficial and additive to each treatment by itself? So experiments started in collaboration with Isabelle Sermet in a patient heterozygote, 3849 plus F508del. We can see here very little residual activity. This is the CFTR function by Using chamber. So here is the result of ETI, and here is the result of treating the cell with SPL84, comparable results. And the combination of these two treatments led to a significant additive effect. More recently, in collaboration with the CFFT lab, we performed experiment in cells derived from 3 heterozygote 3849 F508del HBE cells. And these are the results. The patients are coded by different colors. So CFTR function was measured by the TEC24. So we can see here the average response or the individual results of these patients following treatment with ETI. This is a CFTR function as a result of treatment with SPL84, 5 micromolar. Here is the response to 25 micromolar of SPL84 and 50. Important for the question that I raised is the combination treatment. So we can see the combination treatment when ETI and 5 micromolar of SPL84 were administered, and there is a clear additive effect. And the additive effect can be seen with the higher doses as well, the 25 and the 50. So this, of course, raised the question, does SPL84 and Trikafta, or more advanced CFTR modulator, have the potential to break the so-called potential FEV1 ceiling effect and have a clinical meaningful additive benefit? I'm raising the question because a ceiling effect was suggested based on modulator results. Here, in what we suggest is to treat patients, not cells, but patients, with drugs that have distinct, different mode of actions. Can this be or provide an additive effect? So time will tell. We don't know yet. And I would like to conclude by saying thank you to all the people who are involved. This is my current lab, and Michal and Nomi are contributing to the projects in CF, and these are the people who contributed along the years to this project. And here is Splicent's team with Efrat Ozeri-Galai and Lital, who is here. And we are very proud of the team in Splicent. And last but not least, our collaborators, Isabelle Sermet in Paris, Martin Menz from the CFFT lab, Christine Baer in Toronto, Eitan Kerem. Without him, nothing could be possible. Steve Rowe, who helped us very much in the first steps of the 3849 project, the phase 1 unit, and of course, all CF centers and people with CF participating in our clinical trials. And thank you for listening.
Thank you very much. Batsheва, this presentation is open for questions. Martina?
Martina Genci, UNC Chapel Hill. Wonderful data. Congratulations. I'm intrigued by the variability. You said that different patients have a different degree of correct and indirect transcript. Now, independent of whether it was homozygous or heterozygous, when you looked at those 70% that were more effectively improving with FEV1, were they the ones that had already more correct transcript, or is that hard to say?
This is a very, very good point. We don't have the RNA to do this, but this is a very, very good point. In culture, we do see some correlation, but we don't have any data to support it in patients. Next clinical trial, this should be taken into consideration.
And I'm assuming you don't have any idea why some process it better and others don't, the mRNA.
So variability among individuals in splicing efficiency is known. It depends on the different alleles of those 150 splicing factors. So each of us has a different mixture of these proteins. So in several patients or individuals, it will work better than in others. This is known. It's not only in CF.
Makes sense. Thank you.
Great talk, Batsheva. So regarding the ceiling effect that you alluded to, you show very well that there is an additive effect of the ISO with the modulators. Do you know what that represents in terms of wild type CFTR function? So when you have the two, those around 15 to 20 microamp, right?
Yeah.
In terms of wild type.
And you can compare it to ETI by itself in these cells.
It's impressive.
Yeah, it's almost twofold, if I can say, 82% difference between the combo and each of them, so it's really very significant.
And that additive effect in terms of percentage of wild type, do you know how much does that correspond to?
I think that the wild type, although may Martin be here and answer, I think it's 20-ish.
Yeah, 25, something like that.
Yeah, so we are close.
Yeah.
If we can achieve it in patients, I can retire.
Thank you.
Are there any other questions? Okay, there's none online. I've got one. So this is really positive, but what happens to these patients after this trial ends? Will they continue on the drug?
No. Right, Eitan? They are not continuing on the drug, right?
The phase 2A trial was 2 months, mainly because our safety data in preclinical studies allow us to do it only for 2 months.
Yeah.
And also we did not determine the dose. There were different doses in this study, so we had to determine which dose would be most effective.
Yeah.
Okay, thank you. And just one other quick one. So obviously this is administered into the lungs, but is there any evidence of systemic spread, or in the longer term, do you have any plans to try and look at systemic effects?
Yeah, in healthy individual, this was looked at, and there was minimal systemic exposure. Minimal, dose dependent, but minimal. Yeah.
Okay, we'll just thank Matt Shaver once again. Thank you very much.
Thank you.
Yeah. So there's loads of seats at the front. We'll still be here for another 90 minutes if people want to come and sit down.
Okay, so our next speaker is Dr. Jane Davies, Professor of Pediatric Respirology, and she's going to be joining us to speak about lentiviral gene therapy, the journey into the clinic.
Thanks very much, Deepika, and thanks to you and Paddy for inviting me. I wasn't actually invited. Some of you might be disappointed not to see Eric Alton here today. He sends his apologies. He was unable to travel to the conference. So you've got me, and you won't get as many jokes. So just to set your expectations right at the start. It's the first and last. So it's my pleasure to give you something of an update of what's been sort of 20 years in the making towards the lentiviral gene therapy program that Eric and I and other members of the CF Gene Therapy Consortium in the UK have been working on, and more recently partnering with Boehringer Ingelheim, who I know are in the audience. I do have some disclosures. Of course, we're collaborating very closely with this particular company, and please take what I'm going to say in light of that. So I thought I'd just start really probably preaching to the converted with thinking about the need for gene-based therapies. And one of my favorite pieces of evidence is this from the James Lind Priority Setting Partnership that the Cystic Fibrosis Trust and other members of the UK community undertook, really in this global initiative to try and find out whether in the modulator era, people's research priorities had changed for cystic fibrosis. And you can really clearly see that this is what's on everyone's minds. It aligns very much with what the foundation are doing. And I don't think really we need to state this more. It's a very encouraging, busy area with multiple different trials all going on at several times. And that brings some challenges as well as some encouragement. One of those that is not in the CF Foundation TDN pipeline is the one that I will be talking a little about today. And I will tell you as much as I can. We're in the trial at the moment, and of course, as I think you'd expect, I am bound by confidentiality, so there will be perhaps certain questions you might like to know the answers to, which we'll just have to wait for a future meeting. But I will be talking to you about our journey towards the clinic with this particular type of approach. So just thinking about the various different basket of things that people call genetic therapies, there's a lot of noise about messenger RNA at the moment. Most of the trials are mutation agnostic. The production is apparently pretty easy compared with what it is for some of the DNA-based approaches. And because it's short-lasting, you can stop if you need to. The safety follow-up is therefore shorter. And it might mean that those people could do that trial, come out, wash out, and then have access to future trials more easily. I think there's also a sense of public familiarity since the COVID pandemic, and people are a little more aware of messenger RNA and what it is. On the downside of that, of course, the short-lasting effects really mean that frequent dosing might be needed, and most of these approaches are using lipids, most of which have been shown, at least in some contexts, to be pro-inflammatory. In the converse on DNA, similarly, mutation agnostic, may be longer lasting, and in some cases may be much, much longer lasting in the case of integrating viruses, as I'll talk to you about today. But that longer lasting efficacy also potentially has some downsides in terms of the mandatory long-term safety follow-up that we have to do for regulatory agencies. There are concerns about the theoretical possibility of oncogene activation, and there may be concerns over whether patients get access to future trials. I'll talk about that a little bit as we move on. And this really encouraging data that Batsheva's just produced, it's actually very hard to come after you. You should have been last, I think, today. Really very encouraging impacts, but an ultra-rare population. We had hoped to take part in that study, but we don't have anyone who was appropriate. So to me, it's quite encouraging that these have varying complementary pros and cons to them. So where did it all start in terms of Eric and mine and our collaborators' involvement? The Gene Therapy Consortium is probably something you've heard him and Ute Griesenbach and other colleagues talk about over the years. It really began in about 2001 when the three groups within the UK that were working on cystic fibrosis gene therapy were challenged to come together and work collaboratively rather than in competition by funding from the Cystic Fibrosis Trust. And we were generously funded by them for many years, and then through a number of other grants, we developed what we called our Wave 1 program. And our Wave 1 program was really based on the fact that at that time, it looked like it was going to be very hard to give a viral vector repeatedly and still maintain some efficacy. So we went with a non-viral vector, and this was a liposome-based approach that many of you, I think, will have heard of. Several different trials, including single dose and then multiple dose, led to the phase 2b trial, which followed patients over the course of a year within a placebo-controlled fashion with monthly nebulized lipid-mediated gene therapy. And the data from this showed that we met our primary outcome in terms of FEV1, but you can see here that the treated group had a stabilization, whereas the placebo group actually had a decline over that time. And in the context of modulators becoming available and our awareness of how much efficacy could be leveraged from CFTR function, this didn't seem likely that it was going to be enough. And so we really pivoted in terms of where we would go next. Luckily, the scientists in the consortium had the foresight to think about this scenario before we got there. So all the way along, we'd had a wave 2 bubbling along. And our wave 2 actually was trying to look at viral vectors and see, could we overcome some of the problems with them? What we were focusing on needing was efficient transduction of airway cells, something that was durable and would give us long-lasting CFTR expression, but really, really critically, something that was repeatable. We've seen quite a lot of trials over the years of DNA therapies where the first dose may have been fairly impactful in terms of transfection, but then second and third, and you lose all of that because of the immune function. We also, of course, needed acceptable toxicity. We needed something that could be delivered topically, and in our minds that was likely to be nebulized, and manufacturing. So sorts of things that normally we don't have to think about it in our normal working lives. And at the time we were thinking about these, there was one virus which the consortium had been working with in collaboration with the Japanese biotech, which was really, really efficient at getting into airway epithelial cells. So this is a beta-galactosidase reporter gene, and you can see that the entirety of this animal airway is stained blue because of this gene transfer. And this was a Sendai virus. And Sendai virus was like super efficient at transducing the cells, but it had a very, very short duration of action, somewhere in the region of 1 or 2 weeks. And there was a strong immune response that was likely related to that. Most importantly, we couldn't give it repeatedly. But it did lead to a thought process which has got us where we are at the moment, and that was to question why Sendai was so particularly efficient at getting into cells, and to focus on these F and HN receptors that are on the viral surface and bind directly to molecules on the epithelial surface. This was nothing to do with me. This was the really clever scientists and the people who know about basic virology and vectorology within the consortium. But thinking at the time, what are the sorts of vectors that could last a long time? Lentivirus really is one of those, seems to avoid the immune system and can get into cells and last a long time, but doesn't get into airway cells, really favors cells of the circulating immune system. But what then happened was a combination of techniques to pseudotype the lentiviral vector, so the machine of the lentivirus, but with the surface coated with the Sendai F and HN moieties, which meant that this potentially had the ability to bind very, very well to the airway surface, but when it got into the cells, it had the potential for very long-lived duration. So I'm going to just talk you through a number of data sets, most of which have been published. This particular slide just shows 2 posters that Ute Griesenbach has presented recently at conferences, work done with Boehringer Ingelheim, really looking at expression levels in a number of animal models. So at the top you can see rodent airways there. And the graph on the right shows the copies of a reporter gene vector RNA ratioed to the copies of endogenous CFTR. This is the control group in red with just the diluent, and then the GFP reporter gene with our viral vector showing very high levels of expression. And you can see also in the non-human primate data at the bottom that we got good levels of expression in various samples from 3 non-human primates in this graph. There was no vector-related clinical observations or mortality or changes in body or organ weight that were adverse. Clinical pathology and cytokine analyses were done, which were unremarkable in these models. And there were some microscopic changes in the lungs of vector-treated animals that were considered non-adverse. So all really supportive in terms of us moving forward. These are also some very nice murine data which look at the different types of cells within the lung that can be transduced with this vector. And what we have here is GFP as a reporter gene. We've then got in the middle panel a number of different stains to try and isolate the type of cell that we're looking at. So beta tubulin faciliated cells, for example. I won't go through all of these, but you can see that in the vast majority of these overlays, you can see some signal from most of these cells. We did see that basal cells were pretty sparse, and that may be not too surprising given that mostly they're not exposed on the surface of the airway. And unlike some other lentiviral approaches, we're not using any adjuncts to increase accessibility through the respiratory epithelium. These are some data that I particularly like because they were data that really convinced us that we could give repeated doses and still achieve expression. And to our knowledge at the time, this was the first of these type of viral approaches that really conclusively showed this. Now, of course, because we get a long duration of signal of our first dose, it would be very difficult to know that a second or third dose had achieved expression. So what we tend to do is use a reporter gene for doses 1 and 2, and then we use the gene of interest, but the vector is exactly the same. And you can see really good levels of expression on the third dose here with no blunting of that at all. And then functional restoration. We had seen functional restoration in a number of other models, but this was a nice organoid model that many of you will have seen. Negative control of CF cells on the left, and then our treated organoids in the middle panel here. And in terms of graphing that out, you can see a significant improvement in CFTR-related organoid swelling. And along with that, in this final band on here, mature C band, and not seen in any of the controls. There's a lot more of these details in this paper in Thorax from 2017. If people are interested in that, it's also got quite a long online supplement with many of these details. We obviously had to spend quite a lot of time looking at manufacturing and how we would be able to tool up for a human trial, and very glad to have partnered with Oxford Biomedica, who's got a lot of experience in this area and has really supported us in this endeavour. And then finally, just a couple of graphs on safety from preclinical models. So what we're looking at here is murine data, 3 different panels, naive, PBS treated, lipid treated, so this was the liposome that had been in our previous multi-dose clinical trial, and lentiviral here. And you can see that lung inflammation scores were actually not as high as we saw for some of the well-tolerated lentivirus in human. We looked at the survival of mice right out to the end of their life, and this looks as though it's only one line because actually those dots are so overlapping themselves. The vehicle-treated and the lentiviral-treated mice, there was no impact on survival. And then the final bit of safety I'm going to show you is thinking about integration site analysis. So this is something people are concerned if we integrate a viral vector somewhere near a proto-oncogene or near and influencing a tumor suppressor gene, for example, that there's a theoretical risk of malignancy in that cell. So we wanted to have a look at where the integration sites were, and what you can see here in the little blue and red markers is that the integration was not particularly targeted in any one area. It was throughout the genome of the mice. And for people who understand this, there's a lot written in the paper. I don't fully understand it all, but the distance that it was from some of the important promoter regions of the gene made it look as though pretty much it was going to be unlikely to cause problems. Now, the other thing that we're also thinking about is the likelihood of the cell type to become malignantly transformed. And I think some of you will know that there have been a number of cases of people treated with integrating viruses for other diseases who have gone on to develop malignancies, in particular leukemias in children treated with severe combined immune deficiencies. One of the things really to emphasize about the way this vector has been designed is that this LTR, which is the very, very strong driving promoter part of the machinery, has been deleted from our lentiviral vector, as it has in many others as well. And so if we look at the history of gamma retrovirus vectors, non-self-inactivating, and then some of the more recent bluebird lentiviral vectors, these have not had that part deleted. And so there's still the potential for this very, very strong promoter region. There have been no malignancies or deaths reported from lentiviral vectors which have had this region deleted. In addition to which, we think that the likelihood of the relatively slowly dividing airway epithelium versus the bone marrow epithelium, which is much more rapidly dividing, probably makes that less likely. But it is, of course, something that we'll be very carefully looking out for as we move forward. So into the clinic. So earlier this year, there was a press release that Boehringer Ingelheim had taken the license on this product and was sponsoring a clinical program going forward for first-in-class inhaled therapy for people with cystic fibrosis. Eric is the global chief investigator of this trial, and as I mentioned, we were partnering also with Oxford Biomedica. So just briefly, this is the trial design. It's a phase 1/2 trial. It's designed to look primarily at safety, tolerability, but also efficacy. We're currently recruiting adult males and females of non-childbearing potential because of the integrating nature of the vector. And we're recruiting people currently who are ineligible for CFTR modulators, although of course, this is an agnostic approach and could be useful for any type of variants moving forward. The primary endpoint is any drug-related treatment emergent adverse event within the first 6 months of the trial. And then there are a number of secondary endpoints, which include looking at treatment response, which is defined as a change from baseline % predicted FEV1 of more than 5% within 8 weeks of dosing, and an absolute change from baseline FEV1 at week 24. Of course, we'll be very, very focused as well on looking at dose-limiting toxicity for the duration of the study. Now, at the end of that study, we're asking all participants to roll over into a mandatory 15-year observation follow-up study. And this really is something required of us, of the regulatory agencies. And if you want to know more about what's in these trials, the NCT clinicaltrials.gov numbers are on there. We've also fairly recently published a short review on how we're going to be tackling some of this. So the trial design is, as I mentioned, a phase 1 period where we're looking at small groups of 3 ascending doses in a single dose fashion. And then we will, after an 8-week interim analysis of the final dosing cohort, go into phase 2, where we aim to look at 2 different doses of IMP versus the placebo group. And then, as I've mentioned, this 15-year open label follow-up. So we're currently at phase 1 of this trial. We started earlier in the year. We have a temporary halt on recruitment while a protocol amendment is going in, and hope that we will be starting recruitment again in the fairly near future. The patients who are in the trial continue to be in the trial, and some of them have actually rolled over into the open label extension. So I'm just going to spend 2 minutes on my views on some of the operational challenges in this space. This is not specific to the lentiviral program, and it really may apply to many of the trials that we're doing at the moment, but I think it's a useful opportunity for all of us to think about them together. It's a very, very congested trial space. The population for whom these treatments are so necessary is actually shrinking with every license expansion of modulators. And within that small population, we will need to see how much enthusiasm for new trials and new opportunities there is from subgroups, many of whom in the recent past have not been very engaged with research and may not feel that they're very involved. And that's something that I think we'll hear a bit more about later. By their nature, early phase protocols are quite heavy, both on the participants but also on the study teams. They may involve procedures which are invasive, such as bronchoscopy, and it might be very difficult for us to open multiple center sites for these sorts of trials when we really need very specialist investigations and procedures. So maybe a hub and spoke model would be the right way for this to go. We're going to need an enormous amount of education and information, not only for the population of people who might be considering participating in our trials, but also for the trial sites and all of the staff there. And as I've mentioned, the safety and follow-up is something of a challenge. That long-term follow-up, I think, poses a challenge for all of us in the CF community. We don't yet know for our trial whether this will have to be coming back to the study sites for the whole of that time, but obviously there are geographical limitations to this. Participants may move away or become lost to clinical follow-up, although that is relatively unusual. Registries, I think, will be our friend in this. They're a massive asset, and many of our countries have got very well-developed national registries that I think we could be utilising in this regard. And one of the things that worries me about many of these approaches is how a future sponsor might feel about enrolling or giving an opportunity to enroll for somebody who's been in one of these genetic therapy trials in the past. I think this is something that's really important. A future sponsor might have concerns over contamination of their long-term follow-up data, whether they'd inherit a previous sponsor's risk, for example. Whether seroconversion might blunt efficacy if there's similarities between the 2 different types of vectors. And also, there may be some concern over access to data. But I think there are things that we can do to mitigate this. I think there are a number of practical things we can think about, and various groups are thinking about that. We do have the ability to test any samples that do arise in the case of a malignancy for integration site, looking for the actual genetic therapy, if it has been involved, we will be able to find it. And I think that's something that's really important. And I think as we look forward, perhaps it is a pipe dream or a nirvana, but to be able to work together as a community and think about sharing some data sets and actually making the most of this very, very limited resource so that we speed up progress, I think is something that's great. So I was very pleased to be part of this initiative, which was pioneered by Nicole Hamblett and others in the TDN, maximising the opportunity for therapeutic success and potential sequential participation of patients in these trials. And so just coming to a close, I obviously would very much like to acknowledge everybody in the Gene Therapy Consortium, particularly Eric Alton, who's led us so ably for many years, also colleagues in Boehringer Ingelheim. But I would really want to thank also the clinical trials networks globally. So the TDN, the CTN in the European countries, and in the UK, the Clinical Trials Accelerator platform has been absolutely fantastic in keeping up energy and pace with the UK's involvement in this trial and many, many others. And I think it's a great illustration of how people can come together and really speed up progress. So thank you very much.
I could probably go if that helps.
Could you come to the mic? Because we've got people online. Thank you. But I agree, you probably could, yeah.
Sorry. Just a couple of questions. I think the first one, I was wondering how frequently you expect to dose for this, is the first question.
So I think the honest answer is we don't know yet. And part of what we will be looking for is if we see an efficacy signal, how long it takes for that signal to wane. And we're going to obviously be taking every opportunity we can to try and then deduce something about the durability.
Okay. Fair. The second question is if I read the graph right, the amount of CFTR made from the vector versus the endogenous CFTR is like a couple orders of magnitude. Does that concern you that overexpression might be an issue?
I think it probably would be good if you had a look at that paper, because there's a number of different ways of expressing how much CFTR is actually being expressed and whether or not that's the vector copy number or whether it's actually CFTR itself may not come across in some of my very brief and probably a little bit scientifically ignorant way of showing the graphs. We have talked quite a lot in the past about, what if we get too much? I think our honest answer is that would probably be quite a nice problem to have. And I think we're likely to be on the other end of the scale rather than that.
Okay.
Yes, I have a question. Humans are not susceptible to Sendai disease, so that people don't get sick. So suggesting there is a very huge natural immune barrier. How do you foresee repeating doses in the immune response to your vector?
Thank you. It's a good question. We have some data which I think are really quite supportive from preclinical. There's a similarity between Sendai and human parainfluenza. And we've had quite a deep look at how levels of immunity to parainfluenza affect transduction efficiency. And it seems to be very, very little impact, if any. And so I think that's probably the best we can get at the moment. From the animal models that we've got, we're seeing quite an encouraging signal. Obviously, the proof of the pudding will be when we do come to give subsequent doses. We're very aware this isn't going to be a one-off forever. We hope that it will give us a lot of duration, but we will at some point need to give subsequent doses.
The second question, the organelles that you showed, they were human cells?
Yes.
Okay, thank you.
Hi, I just wanted to, Am I live? Okay,
Yeah.
thanks. Hi, I just wanted to ask if, I know you probably can't speak to the number of people going through the trials currently, but I was wondering if you had any issues with enthusiasm for a trial like this, given that you're working with, viral vectors, and of course, the COVID vaccine here in the United States, at least, has become a little bit controversial, right, around a misunderstanding of how mRNA works, which you touched on a little earlier, but also just in general, we've also had some trials a long time ago, right, with viral vectors that didn't go too well, and that can sometimes make people weary. So I was just curious how you were addressing that educationally, and then if you had seen any enthusiasm for this trial so far.
So we've taken quite a number of opportunities in the preceding period before the trial of talking to the patient community and the parents of young people who might be getting close to adulthood, etc. So you're quite right. I can't talk to you about the individuals we've approached to potentially participate, but I can tell you that more broadly in the population, there's a real spread of opinions, I think is the honest thing to say. And in a way, it's quite good that the trials networks have multiple offerings. So if we're doing an mRNA study as well as a viral gene therapy study, for example, people can sometimes weigh those up. It's not looking like being a really big problem at the moment in terms of the level of enthusiasm for a trial which is relatively small in numbers. But I think as we go on, that will become clearer. And I think you're right, education is really, really key, because if people are mixing up in their minds, like the old story of the adenovirus or more recent anti-vax stuff, we really need to address that with facts, I think.
Thank you.
It was a great presentation. Thank you. Just a question about the powering of the placebo-controlled part of the trial. A 5% change in FEV1. There's a lot of variability in that measurement. Are you worried at all that you'll have an N that's too small, really, to see a lung function improvement, and then have to fight an uphill battle after that because of the way you've designed the study?
Yeah, I think that you're absolutely right. And in this type of phase of a trial, we're balancing everything in terms of saying, can we see enough to continue to progress with the program versus not using up everybody and every resource that we've already got. It won't be the last trial, I'm sure.
Thank you.
Thank you very much, Jane. It's my pleasure to introduce our next speaker and my co-moderator, Dr. Patrick Harrison, who's going to be speaking about gene editing in CFTR.
Okay, thanks for the introduction, Deepika. I'm in charge of time. We'll be fine. The title of my talk is Editing the CFTR Gene, Less is More. No disclosures, except I didn't do this all by myself. So there's a ton of people in Cincinnati, where I've just moved to in the last 18 months. There's 4 people from Cork who are in the audience, so I'll be directing the questions in that direction. A lot of collaborators in the room, lots of different places. I've just met Paola for the first time at this meeting a few minutes ago, which is great. And obviously a lot of people who've supported this work now for many years in terms of funding. The title of my talk, Less is More, you can see I've shortened it already, is a homage to this architect from the 1960s and a more minimalist style of architecture. The real inspiration for my talk, though, is one of Seattle's famous overthinkers who came up with this modification: if less is more, think how much more could actually be. And I think this actually represents what we're doing in the gene editing field sometimes, of trying to make it a little bit too complicated. If we're doing this in the lab as an academic exercise, this is absolutely fine. But if we're trying to get this into patients, maybe we're making things a little bit too complicated. Talking of more, less, this is the total number who've been successfully treated with a gene-based therapy or a gene editing strategy. And what we've already heard about is getting to this number, which is 1, that proof of concept that we can really do this. There's huge efforts. There's far too much in terms of delivery, legal issues, recruitment issues. We'll hear more about this from Deepika in a little while. We've heard a lot about it already. So my talk is about how do we get there and how do we go further. In terms of editing, let's just consider the top 10 variants. And I think Jane just mentioned that paper from Michelle Hamblett and colleagues who have really focused in on the fact that these gene-based strategies are going to be targeting people who don't respond to modulators. So if you look here in this first column, we've got 7 mutations which we could fit into that category of modulator non-responsive. If we look at base editing, I'm slightly biased towards this for reasons I'll explain in a moment, there's only 3 that we could really do very efficiently. And if you look a bit further, there's only 1 of those variants which we can edit without a substantial bystander. If you then adjust for the fact that there may be 1,800 alleles for R553X in the CFTR2 database, most of those people will have a second allele which will respond to modulators, and hopefully those people are taking modulators and doing very well because of it. So the actual group of people with R553X and no second modulator responsive allele is actually very small. Now, that's not enough to make a CF gene editing strategy commercially viable, but I think it's more than enough for a proof of concept study and more than enough for a non-commercial sponsor to potentially take this on if we get there. It's not news that R553X has been targeted by base editing. David Liu and Paul McRae published the first paper in 2021, and there's been 4 or 5 other studies where a lot of people have done a lot of work on this in terms of delivery methods. You can see the whole variety of options here. Just to remind you of the online conference, 5 years ago this week, Hilary Valley and myself from the CF Labs recruited 4 speakers, and one of them, Fedor Urnov, made this statement: the only way to find out how to edit people is to edit people. He's not encouraging us to be gung-ho. We're doing lots of safety studies, or lots of other groups in lots of different mouse models, primate models. But if we really want to do this in people, we have to take the plunge at some point and do this in people. So what's the best route to do this? Well, we said we need to get to number 1. That's for other people in the room. My remit today is how do we go beyond 1? And that number's quite big, depending on where you look. From the CF Labs and Martin's group, there's over 655 CF-causing variants which have been identified. That list has gone up to over 1,000 in the CFTR2 database. We know from the original CF database that there's over 2,000 variants, many of which still haven't been fully characterized. So how are we going to do all these? Well, as we've heard already, the traditional way, and it makes complete sense, is to go with cDNA, because cDNA was shown back in the 1980s to be able to complement any genetic mutation. So if you could deliver this effectively, you could correct all variants with one drug. This would be great if we could do it. The other option, which as far as I became involved in this field, occurred in 2005, which was the development of zinc finger nuclease gene editing. But the limitation traditionally has been that you can only do 1 variant at a time. So back on Thursday, June the 2nd in 2005, I was sitting at my office in Cork and I read this copy of Nature and I read this paper twice, showed it to my student Rowan the following day, and we set about trying to see if we could apply this to cystic fibrosis. So how do you edit a genome? Well, it's actually very simple with zinc finger nucleases. There's just 5 steps that you need to consider. So if we look at the wild-type genome, this is just a small part of the CF gene, and the mutation which occurs in most people is 3 base pair deletion. We can use our mini gene editing keyboard with 5 functions. The first, and it's truly quite remarkable, is to find that mutation. Now, we know where it is in a database, but to find it inside a cell is really quite a remarkable task. But that's feasible with zinc finger nucleases. Step number 2 is to cut out that region to make a double-stranded break. Then press Control D and introduce a donor sequence, the correct information that we want, copy that, paste it into place, and off we go. That took me a minute. It took us 7 years to do that in the lab. But it was done and it provided a really useful proof of concept. And one of the big problems was working with zinc finger nucleases. Luckily, as we all know, within a month of that publication, CRISPR came out, and I remember talking to Mitch Drum in Malta in 2014, and we were discussing experiments, and we were both just about to say that could take us a year, and we're like, we could do that experiment next week. And this is where CRISPR really has revolutionized the research. Students, mentors can come up with ideas, and you can start testing them almost straight away. Jeff Beekman's group was the first one to apply this to CF, and what you're seeing here are these organoids we've just seen in the previous talk, On the left, we've got wild-type organoids, which are swollen up. In the middle, we've got F508 organoids, which don't respond. But on the right, we've got organoids which have been edited, and you can see they swell exactly the same as the wild-type. And that's because they are wild-type. They've been precisely repaired. We know that gene editing has been used for other diseases. I had the opportunity to meet Victoria Gray a few years ago. She was the first person cured of sickle cell by gene editing. Many of you have seen this young lad here, KJ, up in Philadelphia, who's been successfully treated. And others may have heard of this larger scale trial by Beam Therapeutics, who've done a phase 1-2 trial for alpha-1 antitrypsin by gene editing. What's happening in CF? Well, one of my talented PhD students, now a postdoc, who's not at this meeting this particular time, has just published a review on all 26 papers which have been described for CF. And one conclusion that you reach there is that the more editing you get, the more function that you get. There's a fabulous supplementary table there I would point you to. A second review we published last year by Joss Murray, who's in the audience. And it's a very simple question. People always say, which is the best editing strategy? Well, the simple answer, there isn't one. There's lots of different ones, as I've mentioned, but there isn't one which will fix everything. So let me work my way through this list. And I'm going to start with base editing. Now, this is a big publication. It was a massive collaboration between 5 different labs. Martina Scallon, who's in the audience, Paola, who's in the audience, Claudio Sorio, and Isabel Serme, who many of you will know as well. And essentially what we did was we took this base editor, combined it with this guide RNA, packaged it into the engineered virus-like particles, which were developed by Samagya Banskota, and tried to see, could we edit the G542X mutation? No one had tried this one before because you can't edit it directly back to a glycine, but you can refer it to an arginine, and David Bedwell had shown 6 or 7 years ago that this retains a lot of function. We mixed these EVLPs with intestinal cells from patients who are homozygous for the G542X mutation and showed that we could get very precise on-target editing, virtually undetectable off-target editing. And in terms of function, we show nice traces here, and we've also done some organoid work here as well. We see a dose-dependent increase in function as we get more editing. I mentioned R553X. Martina's given me permission to show a few slides which her and 2 of the people in her lab have just performed very, very recently. So with the G542X, the editing was only around about 8 or 9%. But she's now made R553X virus-like particles. And the results here are truly quite remarkable. Even the very low dose, she's got about 60% editing here. And this is precise repair with no bystanders. And with a larger dose, she's now essentially getting complete correction of this codon. So there's a number of experiments that Martina, I'm sure, will be happy to talk about later, if you want to ask her where they're trying to go with this. But that's just one mutation. What about prime editing? Prime editing is very exciting because potentially you can make any correction, any small edit, and you can potentially correct more than one mutation at a time. And this is what we're trying to show in this study, which is a collaboration with people in my lab and Niraj Sharma from Johns Hopkins. It looks a busy slide, but the 2 points I want to make here is we've got 2 mutations, R1162X, which you'll have heard of, and 1158, which is much rarer. This is a strategy to correct 1162X. The slide hasn't changed. This is also a strategy to correct 1158X. With prime editing, you can edit about 30 base pairs, and if 2 mutations are close together, the same editing machinery can correct both mutations. And that's just shown on this graph here that we get a reasonable level of this. Now, this may seem fairly obvious, but Emma, who's the first author on this manuscript, went back through the 4 papers which have been published, and no one else tried to test this idea. So we've just drawn up a list here, and an obvious one when you're looking to correct delta F508 is to maybe see if you can do I507. And again, if this was advanced to a therapeutic approach, if you could correct any one of these mutations on the left, then potentially you could correct a small number of other mutations on the right. It won't be a huge number of people, but for those people, it could make a huge difference. Well, let's just talk about this number, n. So in base editing, it's 1. In prime editing, it's a few. How can we try and get to this much larger number? Well, to understand this, we need to have this acronym. So I'll explain what it means in a moment. I need to talk to John Lewick later on how best to pronounce it. It stands for whole exon editing and beyond. And this was an idea that we came up with a few years ago, and I spoke to Gary Cutting and Karen Rare and the CFTR2 team. They analysed the CFTR2 database and basically put together this table, which is the top 6 exons which have got the most number of individuals with these different mutations who do not have a modulator-responsive second allele. So if you could come up with a strategy to replace a whole exon, and you could do it for all 6 of these, that would be 75% of the people who are currently not eligible for modulators. And a second observation which Gary made was if you could do 11 and 12 at the same time, that would be 87% of the entire CF population, at least according to the current CFTR2 numbers. So how do we do this? Well, normally I'd go down this route of more homology-directed repair XL, which is a strategy that we're pursuing, template jumping, which Joss has reported at this meeting previously. We've heard about super exons by homology-directed repair from Schroem's group yesterday. We're also trying to do this by a slightly different method called HETI. But the title of my talk today was Less is More. So let's do this a bit more simply, maybe. This is a Cas9 nickase. What are we looking at here? In black, we've got the DNA. We've got the PAM site, which is a 3-nucleotide sequence which anchors the Cas9 molecule in the first instance. It's guided to its specific site, in this case, not just by a guide RNA, but importantly, an allelic-specific guide RNA, so it'll only bind one strand. And this is a nickase, so rather than making a double-stranded break, which can give us some side effects we don't like, this will just cut one strand, and we want to see, can this help us? I also want to show this table. So this is the top 10 again. I've taken out the modulator-responsive individuals. But if you think about this in terms of compound heterozygous genotypes, about 99% of people in this category have compound heterozygous genotypes. And you'll see why this is of particular importance in a moment. Also, if you look at these numbers, you'll see it's a vanishingly small number of individuals who would not benefit from a strategy of using a nickase with a compound heterozygous genotype. So what is a compound heterozygous genotype? Very simply, one mutation in one exon on one allele, one mutation, a different one in a different exon on another allele. So all of the information to make a functional CFTR protein is in that genome. It's just in the wrong place. So can we copy the information from one allele to the other? Well, people have discussed this for years. It does happen at excruciatingly low frequencies spontaneously. But we spotted this paper 2 years ago saying that NICs can promote homologous recombination to correct heterozygous mutations. Translated into English, you can use a nickase with an allele-specific guide RNA to cut one strand. That will then lead to the copying of the information off the other strand and repair that mutation. And then you've got at least one wild-type allele, which is enough to bring about carrier status. So can we do this? Well, we can't, but Joss Murray in my lab came up with this mechanism to allow us to see if we could. And this is a fluorescent reporter cell model to understand this process of allele conversion. It's a bit tricky, but bear with us. We made 2 plasmids, one we call B for blue, one we call G for green. And I'll explain this reporter in a moment. But we didn't want to do this with plasmids. We wanted to integrate these into cells. So the first thing we're looking for is a cell which is green and blue, which means we've got one of each of these integrated. It's integrated specifically into the AAVS1 site, and the sequence data here confirms that we've generated the cell line that we want. So what are the details and how do we use this? Well, if you look at the top one there in blue, this is what we call the B allele. At the far right-hand side, you've got a CMV promoter driving the expression of a blue fluorescent protein. But you've also got 2 other fluorescent protein genes here, GFP and mCherry, green and red. But the GFP has been disrupted with a 4-base pair deletion, and the mCherry in this one is functional, but because it's a 4-base deletion, which will give you an out-of-frame shift, the mCherry doesn't work. In the lower one, the G allele, this has a functional GFP, but the mCherry now has got a mutation in it, which means it doesn't work, and also the blue fluorescent protein has a different mutation, so that doesn't work. So how are we going to use this? Well, we did 3 experiments. Joss devised 3 specific, allele-specific guide RNAs, one for the green, one for the red, and one for the blue. And in all these experiments, he's just trying to see, can he restore the expression of the mCherry? So the little green triangle there says that we're trying to target this 4-base pair deletion. This is a nice target because you can have a guide RNA, it'll bind that strand, it's not going to bind the other one. This should lead to copying the information from the GFP, which should restore the GFP expression, so that will be edited. But remember, we're not looking for GFP, we're looking for cherry. If we have restored that frame shift, then now we should see the cherry expression. So what happens? So the workflow is very simple. We tested with the Cas9 nickase, we also tested with Cas9, we tested with one guide RNA, in a few cases we had a second guide, which I'll mention later. And then we can analyze this by microscopy, FACS, and sequencing. So this was our first set of data. Let me talk you through this. If you look at the FACS traces, you can see that both with Cas9 treated cells or the Cas9 nickase, with this allele-specific guide RNA targeting the GFP deletion, we get expression. We can quantitate it here, about 10 to 15% of cells. It's a very big difference from the transfection and the controls. But what's actually happening here? Well, we make the nick, we take the information from the G allele, that copies over and edits the GFP, that restores the reading frame, that leads to the mCherry. Now, I then said to Josh, very politely, we need some sequence. Josh recalls that event slightly differently. Let me explain what's going on here. It's a little bit tricky, but not that much. What we used was short read Illumina sequences. So these are only 500 base pairs. So we can't do the two at once. That's a limitation, but we'll come to that later because we've got a solution for that now. And then we looked using CRISPR-2 to analyze the different reads. What we saw when we did this is that alleles scored as G, so that's with the functional GFP, have now increased by about 15%. So we're definitely getting repair at the GFP. And alleles scored as B have now dropped by around about the same number. You can also see those speckled ones there. So some of the reads can't be decoded by CRISPR-2. We can do that ourselves, but it's a bit complicated. But they've just picked up some other indels or SNPs. What Joss also did then was to look at the mCherry, and this was very interesting. So the mCherry, which we shouldn't be targeting, has actually gone down slightly to the wild-type sequence. And what we're seeing is that we're now getting alleles scored as G. So what we think is happening is that we're getting longer repair tracks. We're not just repairing the short region in GFP, which occurs very efficiently, but sometimes we're getting much longer repair tracks. And we're estimating just from the numbers on this could be at least 2 kilobases. So that could be really useful for trying to correct more than one mutation at a time, but we need to be careful because we may be introducing deleterious SNPs. The second one, if you didn't follow the first one, this one's a lot easier. Now what we're trying to do is to see if we can repair this mCherry directly. So we've now got an mCherry allele-specific guide RNA, which will cut the bottom one. That should copy the sequence over, and that simply becomes edited. Again, you can look at the FACS traces, you can look at the graphs, we're getting very nice levels of editing. So again, look at the sequence data, we now get direct evidence of allelic conversion. That's when we look in the red, where we've edited. But when we look over at the green, again, we can see that the GFP now has gone down a little bit. That's because we've got some, not as frequent, but we are getting some longer editing tracks, which again, could be useful, but we just need to keep an eye on this because in some circumstances, that may introduce something that we don't want. Finally then, we looked at the editing of the blue region. So again, we've got a blue allele-specific guide RNA. We wanted to see if this could restore cherry. We're not trying to look for a gain of blue function, we're just seeing what effect it has on cherry. Now, what you might expect here is that we'd edit this and there'd be no impact on the cherry. And at first glance, there isn't. But if you look at those last two bars, they're not zero. So again, what we think we've got evidence of here is that we are getting some longer repair tracks going across and restoring the mCherry, but we've not done the sequence data on that just at the moment. What Joss has now done is taken advantage of Plasmidsaurus, who most of us just use for sequencing plasmids, but they use Oxford nanopore technology, so we can use long-range PCR, and we've now got evidence for the first time of the creation of genuinely wild-type alleles. So we know that we've phased this, that we've repaired both the GFP and restored the mCherry in these situations. Now, I've got a minute left to explain the actual editing of the gene, and this work has been done by Lisa, who is in the audience. A few shout outs here to the CF labs who've been really helpful in giving us cell lines. That's through Hilary and Martin, and Andre, who's been giving us a huge amount of sequence information. And two other people to acknowledge on this slide, Amar, who's a brilliant bioinformatician at Children's, and Kieran Lee, who was the first author on our original zinc finger paper, who's now a junior professor back in Cork, and is helping us on this project as well. So Lisa got all this data. You can see on the far left, you've got the location in terms of the introns or the exons. And Lisa was looking for SNPs initially, which either create a PAM site or destroy a PAM site. And she's focused in on this one in red. And if we zoom in here, we can see we've got a compound heterozygous genotype again. So the one on the bottom doesn't create a PAM, the one on the top does. So again, we can make an allele-specific NIC with an allele-specific guide RNA. And the question is, can we copy over this A from the bottom to the top? Because if we can, we're now editing the CF gene. So reasonable numbers here, about 4 to 5% editing from 3 independent experiments. We are editing the CFTR gene. But that's just converting a wild-type gene to a wild-type gene. I then contacted Hilary, who'd made the 16HB cell lines, which we all use. And I surmised that in the production of making these cell lines, she probably had some compound heterozygotes, and being Hilary, who's extremely organized, she'd be able to find them, which she did, and she very generously shared them with us. So now we've got a delta F on one allele, wild type on the other. Delta F, as you know, is a 3 base pair deletion. We just took the guide RNA from the original Schwank paper from 2013 from Jeff Beekman's lab. And again, we've now showed that we can restore the wild type sequence in these cells as well. IB3s are a cell line which many people have used. There aren't many compound heterozygote cell lines out there, so if you've got them, please do let me know. So this is a F508 del on one allele, 1282X on the other. Again, we've managed to correct this. Again, it's quite low, but I'll come back to that on my final slide. And we've even done primary nasal cells just in the last 2 weeks. And John Brewington's lab, John's in the audience, and 2 of the guys at his lab are here, and Joanna actually did these experiments. So we've now shown that we've done this in primary human nasal cells as well. Still relatively low efficiency, but I'll come to that on my final slide. So I'm rapidly running out of time, but this is my last slide. Less is more. We just need a nickase with an allele-specific guide RNA. At the bottom here, I'm saying that this is compatible with all delivery mechanisms. So if we can get this to work efficiently, you could deliver this as a ribonucleotide protein and a guide RNA. You could deliver it as an RNA molecule and a guide RNA. You could put it into a plasmid. You could deliver it with LNPs. You could even make it as a virus-like particle. You could clone it into any of the virus vectors which are currently in clinical trials. And it's simple because it's just 2 components, and all it's going to do is make a single-stranded NIC. One of the things that we've seen already is that we can get repair tracks which are quite long, possibly a couple of kilobases. So potentially we could correct any exon in CFTR. The longest one is exon 14. All of the rest are about 200, 300 base pairs. We don't know quite how long they'll be. And an interesting question that Chris Penland asked recently is we don't know how short they can be, which leads into, are they directional? Because if you've got a compound heterozygote who's G542X on one allele and R553X on the other allele, you wouldn't want to correct one and simultaneously insert the other mutation. So if we can see if we can make these directional, maybe it could work even in individuals with those genotypes. What's the mechanism? Well, we've been doing a lot of experiments and we've got a lot of ideas to test this. And more importantly, how can we boost efficiency? In our reporter gene, we're getting around about 15-20% editing. In primary cells, it's still down at 1, but that's where prime editing started out, and we've had reports of that moving up to an excess of 50%. And most importantly, I just made some very simple predictions from the CFTR2 database as to how many CFTR genotypes could be corrected, but I'd really like to look and get some real information on those numbers. I've run over a little bit, but I'll finish there. Thank you very much.
Presentation's open for questions. Please use the microphone and introduce yourself.
Hey, Patrick.
Hey, Greg.
Good to see you. Fascinating talk. More and more I want to work with you. The challenge again, like from the delivery perspective, we have all these different editors, and yet when we go in vivo, we still are uncertain because the efficiency is pretty low, even with prime editing and all, So how do you think if there is a way to improve on the cargo side that might correlate to in vivo efficacy? Is there a sort of like why that, discrepancy is, if all things considered, right? Like if the delivery is happening, what are the challenges that still can be engineered on the cargo side that might help in improving in
Yeah. One of the well-known limiting factors for CRISPR is the delivery of enough guide RNA. So if you don't have enough guide RNA, you won't get very efficient editing. And we've got some evidence that we've got some limitations, certainly in the primary cells, that we need to try and boost that. Another one we've used a couple of drugs. I think several people have mentioned these, AZT and ART. Someone help me out, AAD?
AZ.
Thank you. Close enough. But people are using these. We've used some of these as well, and we can see improvements with some, but not all. And again, if that was translated therapeutically, it may be a case of when you're delivering the editing strategy, you may need to put people on a medication for 2 or 3 days, just as a short-term thing to try and boost the efficiency. In terms of the cargo, I think the important thing is to try and reduce the size. And by using a nickase with a single guide RNA, that's about as small as we can get it. And maybe it doesn't need to be Cas9. There's a lot of smaller nickases out there, potentially zinc finger nuclease nickases, or zinc finger nickases could be engineered, which should be way smaller again. So the smaller we can make it, if we just need a nick to stimulate the process, maybe that could be enough.
Thank you so much.
Really very nice work. If I understand allele conversion correctly, it doesn't require cell division, right? So this can happen in dividing or non-dividing cells?
I believe that's correct, yeah.
So that's an added bonus.
Yeah.
We're just a little over time, so we'll wrap there for questions, but we do have opportunity at the end for more. Thank you, Patrick.
Thanks. Okay, so to close out that session, it's now my absolute pleasure to introduce Dr. Deepika Polanini, and she's going to talk about some of the real-world challenges of trying to bring some of these therapies to the clinic, not just for a few people, but ultimately to everybody with CF. Thank you, Deepika.
Okay, so I just want to start by saying that it was really a pleasure to organize this with Patrick, and then as we were planning this, he was thinking, it would be really lovely, DP, if we got through talking about these 3 unique strategies and then someone could talk about what the perspectives were and some of the challenges that are going to be present getting these actually into the clinic. Do you know anybody at WashU that might be able to do that? And I sincerely said, I've only been here 3 years and I'm still learning the campus, but I know we have a new gene therapy center that's coming together. And he finally had to say, DP, I'm asking you to give this talk. So I'm sorry you're stuck with me, but here we are. This is the NACFC presentation disclaimer. My views do not necessarily represent those of the Cystic Fibrosis Foundation, and my disclosures, which are not pertinent to the presentation today. I think everybody in this room is very well familiar with this slide demonstrating the increasingly lower percentages of people who are waiting for their highly effective treatment strategy on par with the efficacy of highly effective modulator therapies, now down to 5% with the recent approval of the vanzacaftor triple therapy. But obviously this is the population that we are critically focused on, including in this symposium today. And I think as I consider this population, I really think about the changing face, the changing demography of cystic fibrosis, starting from the 1938 first descriptions all the way to some of these most recent representations, the final one here being from the 2022 North American CF Conference, and one of the first times that I think that we saw on such a wide platform the demonstration of elderly patients with cystic fibrosis, the demonstration of multiple patients of color with cystic fibrosis. And so I really want to say that we're addressing an increase in age and a changing face of diversity. And while that's a beautiful thing, it also poses some of the challenges and the perspectives that I'll talk about today. So going through a few of the challenges that we need to consider, I really think that some of the reason clearly that we're able to get to the point of diagnosing more persons of color is because we have more unbiased detection. So one of the challenges that has been in the way has been underdiagnosis of the true demography of the CF population. and that includes newborn screening and systematic bias issues. So this paper actually is speaking towards the targeted mutation screening panels that were used in past and the systematic population bias and detection of CF risk. And that's clearly a factor when we're really focused towards targeted panels that are based heavily in European ancestry. Some people in the room may know, but most probably will not, that in the state of Mississippi, the newborn screening panel, if positive for a serum IRT, would then reflex to genotype testing for only the delta F508 mutation. So you can think about the number of people that were left out in terms of having a confirmed CF diagnosis and the bias that existed there. I think the really remarkable thing is that with the advocacy from the Cystic Fibrosis Foundation and in state support, that's actually come to change, but only in the last 2 years. So this has been going on into very recent times. Clearly, the impact and accessibility of full CFTR sequencing is really improving that, but we also have international considerations for the same, and CF is likely underrecognized and underestimated globally. Grace Paul unfortunately had to leave the room, but this is really her work and the credit goes to her for the molecular profiling here in CFTR of 155 total patients, 120 whom had full mutation analysis. And in this cohort from India, approximately 30% had a delta F508 allele. However, 70% included 55 other variants, and I apologize that it's challenging to see, but I will actually read to you here that this second highest bar on the frequency graph is the 3849 mutation that Bathsheba was just speaking to, which really then changes our lens about where these nucleic acid-based therapies may be applicable, clearly in more diverse populations than we might have originally estimated. Furthermore, this figure also shows that there's regional variation. India is an extraordinarily diverse country, and so you can see that the frequency graphs that are shown here look quite different here, the top bar still being the delta F508, but really suggesting that the regional variation in CFTR mutations could lead for targeted mutation panels in India to lead to bias even amongst those regions, and underscores again the ability to detect genotypes agnostically through full sequencing to the best of our ability. This takes me into an opportunity to really share this slide, which was first shared with me by Dr. Jennifer Taylor-Kauser. And it is remarkable the decades that we went on to really discuss cystic fibrosis as a white disease. When the first description of this, formally from Dorothy Anderson in 1938, clearly discussed that this was present in, using a language of the time, a Negro from parents of a child that came from Puerto Rico, Italy, Germany, Ireland, various parts of the United States, but explicitly stating that the racial and geographic distribution was wide. So I think this just illustrates and highlights the importance for us to be able to look at CF as a race-agnostic disease. But there's also an importance in us being able to discern when we're looking at racial diversity versus ancestral diversity, and that is very critical for us to be able to develop equitable treatments for people with CF. So I'll just take a minute to refer you to this review paper, which is really nice, and goes through the principles of race being a social construct that is still quite valuable in health research and can perpetuate, where this can perpetuate the belief of biological differences between races, but clearly there are social factors that impart effects on health that need to be better understood, and so we cannot ignore race. On the other hand, there are times where we should really be considering ancestry in lieu of race. These are two uniquely distinct aspects. Ancestry being a common line of geographic, genealogic, or genetic descent. And we do know that genetic studies are disproportionately conducted in white European populations, hence also why we've had targeted panels that have told people that they don't have CF when in fact they simply don't have European CFTR mutations. And so ultimately we do need greater ethnic diversity among genetic study populations that are disproportionate to ethnic groups' disease burden, and I'm going to spend a moment just discussing that. So this really becomes applicable in my thinking when we're also talking about gene editing approaches amongst CF gene editing strategies, some of which Patrick just outlined. Some of the aspects that we also think about are super exon insertion, such as inclusion of exons 2 through 27 into a target intronic region. And one of the critical aspects is thinking about conserved regions of safe harbor. So I'm always anxious about people's safety. And so I think it's very interesting and important that we're familiar with this paper from Luca Pinello at the Broad Institute that I've cited here that showed that in sickle cell disease, there were race-based associations for off-target effects in gene editing strategies when they tested 2 therapeutic candidates in trial in vitro. And that was particularly in people from Finnish descent. But I had the privilege of having discussion with Hilary Valley and Martin Mentz and Andre in the room about thinking about seeking conserved regions for safe harbor, but equitable presence of those amongst race. For example, if you could find an efficacious strategy, what is the guarantee that that region of safe harbor is going to exist in a white, non-white population similarly as it would in a white population, and you can deliver accordingly. So in my lab, we decided to start looking at this, analyzing genomes from the NIH All of Us database. We identified 307 people with CF and looked at their genomic and demographic data. These are medical record diagnoses of CF, and the CF was confirmed by identifying 2 pathogenic variants in CFTR mapped to CFTR2. And I'll just give you a very high-level look at some of the data here with regards to the associations of what we saw of SNP variation relative to race, ancestry, and to 2 CFTR variants that we're well aware of. So the question that was posed initially is, what is the variability that we might see in intron 1, starting, for example, relative to race? And so when we look at this principal component analysis, Panel A shows you the variation that we see in intron 1 at a SNP level based on race. And actually, there is no significant clustering that can be identified. But I'll also point out that we have 206 white participants here, 35 Black or African American, and very small numbers after that, with the largest then being those that did not indicate or skipped answer. We then decided to look at ancestral variation relative to SNP variation in intron 1, and we really did not have much conclusion there. But again, you can see that there's 187 of European ancestry and 37 of African ancestry. What we did notice is that when we looked at principal component analysis relative to the very common delta F508 allele in this cohort, we actually could see separation and clustering, indicating that we do have SNP variation in association with that genotype. Even more so, we saw that this is present for the V470M variant, which is going to exist obviously much more widely, even beyond those with the Delta F508 variant. So we went on to do full analyses of predictive models of CFTR intronic SNPs and expression quantitative trait loci, or those areas that may be disturbed and lead to untoward consequences in gene expression, to better identify regions of safe harbor, and, for example, the inverse of the integration site analysis that Jean was just speaking of. But I think what this really speaks to is that those regions of clustering that we identified were more related to the molecular biology of the CFTR variant than to either race or ancestry. However, the issue is that we don't critically know right now if that is actually the case, or if we're just underrepresented in sample sizes to draw conclusions based on race and ancestry. And that brings me back to the need to have early and equitable diagnosis. We don't only need to have equitable diagnosis to be increasing those numbers and treating people early and appropriately for disease. We also need to be thinking forward in our new diagnoses about building trust in these populations. So one of the factors that we still need to be able to overcome is when we're thinking about research hesitancy in minoritized people. These are 2 cases of research ethics breach that have been vast and incredibly deep in U.S. history. The first on the left is the U.S. Tuskegee syphilis trial, which was a 40-year ongoing study supported by the U.S. Public Health Service. Tragically, these men were told that they were having their syphilis studied and treated when they were actually being followed despite not being told in some cases that it was indeed syphilis that they had. Once it was found that a simple effective treatment of penicillin could treat these individuals, they went on to be treated studied even longer with invasive procedures, including lumbar puncture, assessing spinal fluid, but never actually disclosed that there was a treatment available to them and given to them. As one simple example of the level of this breach, this study was planned as a 6-month study. It went on for 40 years following these men, and reparations and apologies were not made even another 20 years after the study was closed until the 1990s. So there is clearly strong reasons for distrust that exist with research in the U.S. in minoritized populations. Another example on the right is the story of Henrietta Lacks, whose cervical cancer cells were taken from her without her consent, then turned into the first immortalized cell line, which went on to inform thousands of experiments in the decades since. Her family was only made aware in the 1970s, more than 20 years later, that in fact, not only were her cells used in such wide experimentation, but also as time went on that her full genome sequence was actually publicly available, causing concerns for privacy for her survivors in her family. Her data from these experiments were also commercialized with no attribution or compensation to the family until this was actually brought to further justice with the Henrietta Lacks Foundation. So this is just a couple of examples of some of the significant ethical breaches that we've faced and clear reasons why we may have issues for distrust with communities who we are now diagnosing and often diagnosing late in life. So this is an opportunity for me to really segue to the wonderful work of Jessica Hudson, who was with the Cystic Fibrosis Foundation. She led the community voice efforts in CF with goals of better understanding research engagement in those that are not benefiting from CFTR modulators and how the communities are receiving research information as well as interest and barriers to genetic therapy and clinical trials. Patty asked me to really talk about perspectives here, and that's what I wanted to share with you today. So these are perspectives that I'm going to share really coming from two cohorts. The first of these is from 23 adults and 4 parents. This was a predominantly white population, 2 identified as being of Hispanic ethnicity, 7 were eligible but not taking CFTR modulators, and 16 were not eligible. She conducted 27 structured interviews, about 45 to 60 minutes long, in 2023, and then analyzed the data with thematic analyses. She also conducted a second cohort, which was done in July and August of 2024, specifically trying to seek the perspectives of people of color, as that was really missing from the first set of interviews. So she conducted 10 additional interviews for people of color who were ineligible for CFTR modulator therapies, and that was genetically ineligible this time, including 7 parents and 3 adults with CF. This was done through community voice, and in this case, 6 individuals identified as Black or African American, 3 of Hispanic or Latinx ethnicity, and 1 South Asian. And none were eligible due to mutation, but you should note that this was prior to ALF-TREC approval. So the feelings represented in community and in research, the majority of participants expressed negative or ambivalent feelings when asked if they currently felt represented. It was very clear that people with CF who were not taking CFTR modulators because of side effects had an even higher percentage of negative comments about feeling excluded or left behind. And many felt left behind and deprioritized, but also expressed hope in research, citing recent improvements in visibility and support. And so I know that these topics have really been coming to rise, and increased representation, discussion, and working groups appear to, at least in this qualitative set of interviews, be making some progress in building trust. But these are some of the direct perspectives. One person saying, we've gone through the stages of grief. We were a big CF family altogether, and now we're not anymore. Another, a tiny little subset of us have had to stay on the Titanic. Everyone else got on the lifeboats and we're supposed to be happy for them. We are, but it's rough, including listening to everyone else celebrate. People in organizations constantly need to be reminded that those not eligible for modulators are still living in desperate need of help and more options for treatment. And finally, we're still waiting for ours. It gets frustrating. They get more stuff while we're still sitting here waiting. And so there's clearly a push for the imminent need for effective therapies. Some of the considerations for participation in nucleic acid-based therapy research were polled, and the majority, in fact, did demonstrate that they would consider participation. Some of the positive factors were really internal motivation, citing that this was really towards the greater good. Most were commenting on relying on the recommendation of their care team. And I know that the TDN has provided studies before that demonstrate that this is the number one compelling factor for most potential participants, is the recommendation of their care team members. There were the beliefs that these types of therapies will improve overall health, that they may be able to stop other treatments, that they wanted to be able to have more detailed information about the study and related research, but that seemed like it was increasingly forthcoming relative to past exposure, and then also, of course, compensation. The primary areas of concern were, as we usually see, those related to trial logistics. This is a population, obviously, that is still really mired in the level of the schedule and complexity of treatments for symptom-based therapy of their CF. So working through logistics of time and travel when you're still trying to manage your complex daily regimen is obviously that much more challenging. Concerns for side effects and safety, impacts on daily life when they're trying to balance family needs or their work. Invasiveness of procedures. This is a population that has been through quite a bit of medical trauma with the level of hospitalizations and care that they've received. Also, personal health status, of course, feeling like they're doing too well to participate in research or that their health is too fragile. And then concerns about eligibility for future studies, such as Jane was speaking to, with long-term follow-up periods that may be extensive. What is my ability to participate in more than one trial? Finally, negative past clinical trial experience or concerns around age restrictions. One of the key differences is people of color who were modulator ineligible tended to be less overall aware than the first cohort, which included predominantly white people and included people who were eligible but had side effects. Only half of the second cohort indicated that they were generally aware of genetic therapies like gene therapies or RNA therapies for CF. Of those who did indicate awareness, they indicated that their awareness was very high level, and some were using research or indications and understanding to be applied, in their case, from other diseases. So this really points to the need for increased community education. Only half of participants in the second cohort actually considered that genetic therapies might be a promising option. So I just wanted to highlight the work here for a moment from the CFF's Genetic Therapies Working Group, which has really taken great strides to try to increase the accessibility of educational information at lay levels towards these communities. And you can scan here to be able to access that, or those are all available on the web. So finally, one of the challenges that are just a few amongst what I've been bringing up here to consider is, as Jane also mentioned, that we are increasingly getting into a limited population. These data are actually provided from the 2021 CFF patient registry, and at that time, prior to Alyftrek approval, we knew that there were approximately 1,600 adults with CF who were ineligible for modulators. That number has since been decreased thanks to the approval of Alyftrek. But when we consider the number of those that might be eligible to participate in clinical trials at maximum bounds, thinking about a lung function greater than 40%, no lung transplant, this was about 1,100 people, of whom there's about 25% that would be Black, Indigenous, or people of color, or identifying of Hispanic ethnicity. So again, this is disproportionately enriched relative to the overall US CF population. So this is an opportunity to really come to close by also highlighting for you what we're trying to do to work within these smaller numbers and ranges and also diverse populations by talking to you a little bit about REACH. This is research expansion to advance the CF therapeutic pipeline for people without modulators. So just to give you an overview of the study goals of REACH, this was really designed as a prospective study to collect clinical outcome and biomarker control data. Also to improve outreach, knowing that we have lesser exposure and understanding towards genetic therapies in these trials for people with CF who are not taking CFTR modulators, by developing a study website and social media, since we know that we needed to use perhaps some lesser conventional approaches than research coordinators well familiar with participants reaching out to engage them. And finally, this is really to highlight the work of Nicole Mayer-Hamlet, Natanya Kerper, and many colleagues who are really setting forth to look at how to use these external control data in partnership with regulatory pathways and with sponsors when thinking about innovative trial designs and working through new regulatory processes. All of this is intended really to increase the trial engagement and streamline the trial designs to support the robust developing pipeline. So in thinking about developing the study, the concepts were really keeping in mind the ideal pivotal trial design for a nucleic acid-based therapy and using that as a guide to try to determine what types of control data we would need and at what frequency to support regulatory approval for a new nucleic acid-based therapy. And so initially, we really thought that a 6-month placebo-controlled trial would be the ideal, with research visits at least at baseline 1, 3, and 6 months, depending on the key primary outcome measure per usual of percent predicted FEV1, but also looking to key secondary clinical outcomes such as quality of life measure and CFQR, and more nuanced measures of lung function as well. Additionally, collecting safety data such as safety labs, exacerbations, hospitalizations, and specialized markers focused on nucleic acid-based therapies to help support those safety profile informations as well. This is an overview of the visit schedule. REACH is now active as an observational study using a 12-month study period with 4 visits. This is occurring at day 0, 3 months, 6 months, and 12. In the core study, we're collecting pulmonary function testing and at home and in the clinic, as well as quality of life measures with the CFQR, again, at home and in the clinic, and blood collections for safety labs, as I was just mentioning. But there are also additional sub-studies, including multiple breath washout and mucociliary clearance, that are shown at the frequency of these icons in the visit schedule to try to support secondary outcome measures that may be more granular or nuanced in lung function. And finally, also a sub-study of immune responses, looking at T-cell responses to CFTR over the study period as well. We did design the study with input from the community, and we continue to work with the community in engaging the overall ineligible or not taking modulators population. So I just wanted to highlight some of the comments from community voice and study design, and really individuals were saying that being able to substitute the data that you have with REACH participants, I think that's groundbreaking, and that was really speaking towards the application of using external control data. There were also comments towards trust. I think so. I think more people would get involved, especially those that have been paranoid or new to the whole thing or just curious about getting into research. This would be a start. I won't read everything word for word, but I will just say that one of the participants said, do a poster or a session at NACFC or something to share that this is happening. So here I am. And last but not least, I really wanted to make sure that I thanked our co-investigator, Dr. Hannah Budwani, who has been critical in helping us both in digital engagement, in REACH study participation, but also with our advisory boards. Those are summarized here. We have a community advisory board with 4 members of a wide variety of ages across them, who are across the U.S. in very different geographic regions, all with very different personalities, informing us their perspectives of what the community sees as these types of studies are trying to go forward, both in genetic-focused therapies as well as an observational study like REACH. And then also a social engagement advisory board. We had our first post from a member go live this week. This is 5 members from across the country, including 2 Spanish speakers when we're really thinking about the population. All came into this with a strong social media presence, and that was the basis of their engagement. And they are starting to put out their own blasts or bursts about why they think that participating in REACH is important to start reaching their own demographics that they interact with most. So I wanted to give great thanks to them. So I've talked about a lot of challenges today, and to wrap up, I'm excited on time. You know, it's not all bad. We're talking about a history of underdiagnosis and newborn screening and systemic bias. But as I mentioned, you know, we've been able to make progress changing things from a single mutation bias in Mississippi to moving into more states across the country that are actually looking at reflexing into CFTR full sequencing if mutations are not identified so that we have unbiased detection, and that's already happening. When we think about assessing racial diversity versus ancestral diversity, I think the conversations and even the challenges that we're facing make these more at the forefront of discussion on our medical campuses. So we're improving medical education, in fact, and I hope also improving research awareness about the nuances of these topics and the fact that we cannot ignore either or assume that the two are in fact the same. I think it's clear that we're starting to build community trust and community engagement. I'm delighted to tell you that in the enrollment that we've had in REACH to date, we are already outpacing in these last 14 months the enrollment of minoritized populations such as African Americans or Hispanic ethnicity individuals compared with a 7-year window prior, including both interventional and observational studies. So we are rapidly outpacing the work in the past in terms of engagement. And I think that's a reflection of numerous aspects of this community trying to build trust. And finally, in terms of working with limited populations, we are really trying to work at innovative trial designs and supporting research engagement in new ways. So with that, I just want to say thank you to the people that are listed here and many more, and I'm happy to take any questions.
Okay, we're open for questions. Anybody would like to start? Can I just ask one in terms of this paper, I think Jane also mentioned at this study, there's maybe between 500 and 1,300 people who are modulator ineligible, depending how you count it. That this is the population that you're looking at, presumably. And how many, or what fraction of that population are you striving to get to? Or would you like to contact everybody?
So,
or have I slightly misunderstood?
No, that's okay, Patrick. I will say, in terms of contacting everybody, that is one of the applications that we're using in the REACH study is something that's really called universal screening. And that is an opportunity to take everyone at a baseline level of their eligibility. In this case, people who are simply greater than 12 years of age or older and not taking modulators, and agnostically consider those individuals potentially eligible and approach them without bias of who we think will or will not be a good trial participant. We then actually, if a site is not approaching an individual, we're asking reasons about why, so we can understand reasons for systemic bias. And if an individual declines to consent into the study, we're actually collecting data about why so that we can understand what the specific barriers are or places that we can make progress in building more trust, including based in specific demographics.
I'll just ask a second question. So just before we started, someone said to me, how long do you think it will be before you have these gene therapies? And obviously they're not just around the corner. So what do people say in these qualitative studies when you're asking these questions? Is it like, how do you feel that this could be 5 years or 10 years or longer?
So I didn't include all of the work from Jessica today. There is an incredible depth of work and information that she received. But I do think it's really important to emphasize that communities that were answering to, individuals who were answering to these questions did make comments of, we're tired of hearing about hope. So I think that there really is a need for having really transparent and specific discussions. It's always obviously impossible to address crystal balls, but I think that setting realistic expectations is really what they're asking for, and based on the responses that she received in those two cohorts.
Yeah. Okay, thank you. Okay, if there's no more questions. Oh, yes, we do. Excellent.
I just had a question about, does the CF Foundation have a set of primers for the less characterized variants, like publicly accessible?
I can't speak to what they have available, but we have several people in the room. I don't know if anyone can, and I'll leave that a moment while I... So we are collecting Buffy coat for consenting individuals. So that's an optional consent because we realize how sensitive it is to be asking vulnerable populations for DNA collection. But it is, I have of the same ilk as what Hillary Valley said, that, when you're looking at one person, you're looking at the one person in front of you. And I firmly believe that until we actually have adequate diagnoses and adequate populations in whom we've built trust to have adequate sample sizes, it's very challenging for us to draw conclusions on the basis of race or ancestry in this population right now. And so that's part of why we're collecting Buffy code in REACH, is to be able to look at unique genetic sequences. As far as the availability of primer sets, I really can't speak to that. But I can say in my own experience and in the genetic therapies working group, I have heard minoritized individuals say, I just don't feel like these therapies were made for me. And when you pause and think about the reference sequences that we're generally working from, that's coming from a general gestalt that may in fact be true. That's
Okay.
not to say that they would not be efficacious.
Apologies there. Okay, it just leaves me to thank our speakers, Batsheva, Jane and Deepika, and wish you a very good rest of the conference. Thank you very much.